blobs. So blobs give us color. Complex and simple cell of cortex. Of what cortex? This striated cortex give us feature detectors, means they can detect minute changes. What about extra striated? The area which is not in Brodmann's area 17, 18 and 19. So this is not in this area, so we call this extra striated or extra striate corticals. So we have two parietal temporal lobes, and I told you magnocellular pathway. M stands for movement, P stands for parvo cells, small cells, and for picture and color, picture perfect, picture detail, right? So this parvocellular pathway is inside temporal, magnocellular pathway is inside parietal. Function, we know, function, eyeball movement, color vision. So if you have any sort of seizure going, what is seizure, by the way? Seizure is just misfiring of neurons. If your neurons are irritated by any sort of chemical messenger or any sort of neurotransmitter, your neurons will misfire, and then you will think that you know someone. You look at their face and you think, okay, I know them, but in reality you don't know them. So this feeling of knowing someone, and it's actually you're going through a seizure, and there's something called as deja vu. When you think that something has happened already but it's happening in real time, this feeling is also under seizure. And this is a seizure. This deja vu is the seizure of inferior temporal cortex. Inferior means beneath, temporal means temporal cortex. So the temporal cortex that is beneath this, you get the anatomy. I'm trying to explain you the anatomy, that's it. So the seizure of inferior temporal cortex is deja vu. So this inferior temporal cortex it works for face recognition. And how it does face recognize? It recognizes face with parvocellular pathway, and that's the parvocellular pathway helps in this face recognition, right? Cool. What is this disease? It's called prosopagnosis. Well, prosopagnosis, weird name. Prosopagnosis, not prognosis. Prosopagnosia. Gnosia. Okay, I will tell you. Prosopagnosia is a loss of recognition. You can't really recognize someone because you have some sort of lesion or some sort of stroke inside this inferior temporal cortex. So this is disease. Cool. What about color vision? Well, you know color vision is done by two things, cones and parvocellular pathway. So pathway, cones, they receive input, and then this input goes to thalamus, parvocellular pathway, and then this eventually goes to blobs. So this is how it runs. We have first cones talk to parvo talk to blobs, because blobs are present in area number 17, 18 and 19. Okay, what are cones? Cones are just these conical shaped cell. They are primary, they detect primary color. There is RGB. So RGB, the wavelength. We know that these cells detect these colors, right? So these cells can be divided, can be classified, types of cone can be classified on wavelength. If it's long, it means it detects red, because red comes under long wavelength. Like if it's medium wavelength, it's green. If it's blue, then it's just S cone, M cone, medium cone, long cone, medium cone, small cone. Small cone detect small wavelength, medium cone detect medium, that is green color, and long cone detect long wavelength. So RGB, long, medium, small cells. Simple. Okay, what about color blindness? This is color blindness. It is a X-linked recessive disorder. What does it mean? Well, it means that in humans we have two type of configuration. Females have something called as XY sexual chromosomes. Men have XX. Sorry. Men have XY, women have XX. So what it means that it's a recessive, right? So in women, if one chromosome is defective, but they still have one X healthy, so they will not, you know, suffer from this disease, suffer from color blindness. But men have only one X, and if this one X is messed up, they will suffer from this disease. And this is the reason males are affected more than females. Cool. What about females? Females are carrier because they might have this defective X, right? They might have this defective X, but they might have this healthy X as well. So they are carrying this disease, but they don't know about it. So X-linked recessive means women are carrier and male they suffered from this disease more. Okay, most common is red-green defect in men. So red green defect, it means they won't see the red color, they won't see the green color. What about the name? If your red is messed up, we call this protopnia. If your green is messed up, we call this deuteropnia. If your blue is messed up, we call it tritopnia. You just have to remember this. I usually remember this by red. Red is the color of blood. Blood is present in all organism, and organism. Blood is like old, right? It was present forever. It has been present forever. So I just think about proto. It means old, and so that's why old. What about deutero? Well, in India there is a soft drink brand. It's called Mountain Dew, and it is advertised in a green color. It's a green colored soda. So Dew, Mountain Dew is green, and blue is tritanopnia. I don't really like to remember this in any way because it's like I can just eliminate these two and then I'll remember about tritanopnia. Cool. Order of neurons. So we have studied this. So what happens that first light comes to photoreceptors, rods and cones. Then light communicates to bipolar cells. And then light communicates to ganglion cells. And then ganglion make axons. These axons are nerve. Once they enter inside the CNS, we call them tract. So first order neuron starts from photoreceptors. Second order starts from bipolar, because I usually remember this with bi equals to 2. So bipolar is 2. Third starts from ganglionic cells, right? Because they make the axons that go into LGB, lateral I always mess up the name. Genital, I don't know, genital geniculate. So lateral geniculate. So ganglion provide lateral geniculate body, which is in thalamus, right? It has 6 layers. So 2, 3, 5. Uranium ipsilateral. Do you remember this? I hope you do. We have 2 cortex. One is striated, second is extra striated. Extra striated has simple, complex, extra striated, sorry. The striated has simple, complex, blob. Extra has parietal, temporal. For colors, first cone, light hits to cone, then it goes to temporal, and then it goes to this 17, 18 and 19. Cool. The final pathway. Let's study the final pathway. So we know about this order of neuron now. Okay. The light reflex. Cool. What was light reflex? Do you remember, guys? Well, light reflex is the reflex of your pupillary muscles. When we hit pupils with light, these pupillary contraction happens. How this contraction happens, I will tell you now. First of all, we have to remember this: pretactal nucleus and Westphal nucleus. This is common in MCQ. Okay. Okay, we are here. This is a reflex pathway. Let's start from 1 to 9. These are 9 stages. I try to understand this way. First, this is the light source. It goes and it hits into the eyeball, and then this optic nerve is, the light reaches optic nerve. Nerve, it's outside, it's PNS. After this light, this photon is traveling to something called as chiasma, where the crossing happens. Then this photon travels to optic tract, here or here, whatever you want to call, it doesn't matter. Here, optic tract, it's here, let's say. Then this light goes to something called as pretactile nucleus. Do you remember? Pretactile, before tactile nucleus, cool. Crossover happens here. What kind of crossover I'm talking about? This photon is traveling here, and this is a pretactile nucleus. Here, instead of going here, it goes here. Okay? It goes here. Crossover happens. Here, what is here? Do you remember something? We call this Westphal nucleus. Sixth is Edinger-Westphal nucleus. So light is coming from here, and it cross over here and then here. It activates on both sides. Okay, so even if you hit eye with one-sided photon and it travels here, it will activate the nucleus on both sides. Okay, this is how it happens. And then once both sides of the nucleus are activated, then the photon moves to efferent, which means coming out. Efferent oculomotor nerve. It's nerve. It means it's in PNS. It's outside CNS. Here. So from here it goes here. It's called oculomotor nerve. It's on both sides. Then it goes to ciliary ganglion. Ciliary ganglion, the axon making ganglion. Here, both sides are activated. Then it goes here. Then it goes sphincter pupillary muscle. Again, the light is going in both ways, here and here. So both of the eyes are activated. Both of the sphincter pupillae muscles are activated. Both sides of the muscle constrict, and then we have something called a pupil constriction. And then the light source, if the light source is present, we call it light reflex, direct light reflex. If the light source is not present here, we call that indirect light reflex, which is normal. If you have problem with one eye, if you have some lesion here, or maybe here, or maybe here, then you will have one indirect will not work and direct will work, or maybe vice versa, depending upon the location of the lesion. So that was it. Not so tough, right? So this was vision. This was your whole vision.